you do before genetics? I think everybody of you did the 1080 right? Before? So you have some idea. Okay. So I am your instructor and I am Palash Mallik and you can call me Dr. Mallik. So, and I'll be teaching this course. We'll be meeting on Tuesday and Thursdays, 11 to 12:20 here. And to call me, actually, I'm not here all the time. So best way to contact me is the email. Okay. So if you have any kind of questions, any kind of problem, just email me and I'll contact you. And if you have something that is like any paper or any kind of project you need to submit later, not right now, later on, you can go there to the faculty service center and you can drop it up there. Do you know where is the faculty service center? It is just by the breeze. There is, you can take the right turn and the first room in the hallway. Okay. So what we will learn from here. So what is genetics? What do you mean by genetics? Do you have any idea? What is genetics? Yeah.
It's what we made up of our DNA. Just kind of
Yeah,
like
genetics is DNA, but what is that actually? The subject, when we are talking about the biology or like DNA, RNA, that means it is sometimes we call this molecular biology. Okay, gene expression, DNA uh, technology or biotechnology, those kind of things. That is called the molecular biology. But what is called actually genetics? What are there? And there are suppose photosynthesis, cellular respiration, those kind of things are there. But in this topic, what we will go to learn. Genetics means mainly the transfer of genes, or mainly the classical genetics means the inheritance of genes. How we look like our parents. That is the question first. So that is our genetics, but that is the classical genetics during Mendelian genetics. So who is Mendel? Do you have any idea?
The father of genetics.
Yeah, father of genetics. But why is he called father of genetics?
He studied pea plants and
Yeah, actually, he is the first person in this world who coined the term that is the inheritance. He didn't coin the term genetics because that time he didn't know what is gene. He called it as factors. But he was the first person who told us that the some factors are transmitted from father to their offspring. That is the inheritance. So we'll talk about this Mendelian genetics a little bit. And Mendelian genetics includes these genetic traits. Genetic inheritance means autosomal inheritance. Autosomal means we have two types of chromosomes in our body. One is called the autosome, another is called allosome or sex chromosome, which determines the sex, that is X chromosome and Y chromosome. So the genes related to autosome and genes related to sex chromosome and how they are transmitting from one generation to another generation, we'll learn about those things. And the mutations. What is mutation? Mutations means some kind of defect in our genetic characters. Okay, that is called the mutation. Suppose Down syndrome, suppose Klinefelter syndrome, those kind of things, even cancer. Some kind of genes are responsible for doing cancer, mutations. Those are the mutations, chromosomal aberrations. Then we'll go, this is the main part, that is the transmission genetics. Then we'll go for the molecular genetics part. Molecular genetics means molecular biology of the gene. So what is gene? Do you know what is gene? A very common question. What is gene? We are talking about gene. This is a whole syllabus, whole semester is about gene. What is gene? Supply. Is a point or something? What is gene?
it's like a piece of your DNA that's
Yeah.
a part partially makes up your whole DNA.
Yeah. more or less close, a little bit more descriptive. So we have the DNA. I know that we have the very long DNA, almost like 6 billion base pairs of DNA. But the whole DNA contains a lot of genes. That means a part of DNA. So a part of DNA which can synthesize a particular protein that is called a gene. So this is the main concept. We'll learn about that one, what is the structure of the DNA, replication of the DNA, how DNA replicates from one DNA to another DNA, then how DNA or gene express, that is the protein synthesis, how they form proteins. That is probably you know the terms like the translation and transcription, those two things. Okay. Then there are some other parts, the developmental genetics. Developmental genetics means how the genes are responsible for developing our body. So from embryo, because we all generated from a single cell, that is called the zygote. From zygote, each and every part of our body developed. Okay. But the thing is that our skin, we have the cells here in the skin, we have the cells there in our eye. But why the eye is not like the skin? We have the same cell. It came from only one cell. Okay. So what are the things there? That means some genes are expressed in our skin cells. Those genes are not expressed in our eye cells. That is the expression of genes. Something is controlling that one. Switch on, switch off process. This gene will be switched on into this part. This gene will be switched off into that part. Okay. That is called the developmental genetics. Okay. Then DNA technology. That is the most famous topic right now. DNA technology. How to alter the gene therapy, mainly popular for cancer therapy. How to alter the gene, even how to produce the modified organisms. When you are going to the groceries, you will find the GMO products. What is GMO product? Genetically modified organisms. That means something that which is not giving too much. Suppose some paddy, and they are not yielding too much in the field. So we modified some genes that they can produce more. They can be longer, they can be thicker, so we can get more from that. So that is the DNA technology. And not only that, some kinds of medicines are produced by this DNA technology also. And the population genetics. What is population genetics? Population genetics means that in this population, one gene is transferred from one to another. We have different human races, different colors. Why? Why are those? Or the new species are formed. We all came from apes. So why the apes transformed into this kind of human? That is the evolution, adaptation, and that is related to the gene. All these things we'll describe in this chapter, population genetics. So clear? Do you have an idea about that one? Now, couple of official things. This is your textbook, main textbook. That is the Benjamin Pierce Concept of Genetics, Conceptual Approach, 6th edition. Okay. And I talked with the bookstore people yesterday, and probably they will get it very soon, if not today, because yesterday I talked with them. So this is your main book, or this is our main textbook, but I will suggest that you can go for some other books also. You need not to buy all those books, but if you find it from the library or anywhere, just study it. And some other old books are there. One is Stansfield's Genetics, that is very good for the population genetics. And another is Strickberger Genetics. If you find this book anywhere, I would suggest just read it. Yeah, job. They took everything from here. So this person, this is a very old book, and almost in 1969 it was first published. So when we read genetics, we use that book. But this is a very, very good book until now. So if you want to learn genetics, I will suggest that you can, if you find this one, you can read this book. Okay, and this is much more advanced than the peers, much more advanced than the peers. But if you have interest on that one, you can go that one. But mainly we will follow that one. Okay, and other materials means just keep your notes here, take notes, and keep all these things together. And probably we'll do some project here. So that is another thing. We have the lectures. We'll do the homework in sapling. Okay, so if you buy this book, when you will buy this book, you will get a code for the sapling, and I'll let you know in the next class or next week that how the sapling they are already right now preparing all those things. So I'll let you know. Then we'll start the homeworks. Okay, homework will be online, so you can do the online and you can submit that one. And mostly we'll go through online. Mostly, I'll suggest that save some papers. Okay. So we have a term paper, either term paper or a presentation. So you need to go. That is a simple kind of project that you need to work in groups. I will give you some topics, and you need to do some research on that topic, and you will present in the class. So that will be some kind of things like a group meeting or something like that. The one person will study one topic, or one group will study one topic, and they will deliver the lecture to the others. So everybody can learn from that one. Okay, and that will be in our last class. So final exam, this is the date for the final exam. If we'll go for the in-class exam, then will you have to come here at 9:45 a.m. and the exam will be until 11:45. But there is a very good chance that I will go for the online exam. And in such case, during that week, so it is on Tuesday. So during that week, between Saturday and Sunday until Tuesday, you will get 2 3 days there to finish the exam. I will give you the online code for the exam, and you can do it from your home anytime. Okay. And we have this percentage of marks. Homework is of 15%, quizzes 20%. We can have almost 3 4 quizzes there. Exams 25% each, and the project is 15%. Okay. So the thing is that suppose we have the 4 quizzes. So quiz 1 2 3 and 4. All these together, whatever the marks it is, if it is 100 altogether, suppose 25 each, then we will get 100. I'll transfer this 100 into 15 and I will get the equivalent marks up to 15. Okay. But the thing is that what all the students cannot understand that do not miss anything. The best thing is that just go through this pace and you will get B and A. Okay. Even if you go through all the pace together, if you do not miss any homework, if you do not miss any kind of quiz or anything, you will definitely get B. If you do the worst thing, and if you do it very good, then A is okay. Because in another class, I got almost 16 A out of 27. Okay. And you have this extra credit. I call this as pace points. So for your attendance here in the class, you will accumulate 2 points per day, and 10% of this total point I will add as extra credit to your final grade. Okay. So your final grade will be on 100 only. Total will be 100. So 15 for homework, 25 for quizzes, 25 for one exam, 25 for another exam, and 15 for project. 10 plus 1, you have 10 extra credit. And this 10 extra credit is for this one. And in exam and quizzes, you will also have some kind of extra credit points, like 5%, 10% marks. So 5, 10, you can get there. And this is the normal grading system of our university. So 93% and above, there is A, there is no A plus here. So that's why I have something written there in your syllabus. You can find that if you get 93% and above throughout the whole semester before the final exam, you need not to attend the final. So try. A lot of people can do that. For this 14A, I got 7 of them who didn't appear for the finals. So you can do that. Attend class on time. I am generally very much punctual. Okay, generally. So I'm a human being. Sometimes I can have the late also, but generally in last 2 years I didn't fail any class 5 minutes late also. Okay. After coming here, you need to fill out this one. This is your attendance sheet, and based on this one, if you forget, that means you will lose that point. Okay. So do not forget that one. Write your name here, sign here, and put your time when you entered into the room. When you came into the university, I don't need to know. When you entered into the room, yeah, that happens. Because last time some people wrote that she came here at 7:45. I had a class at 9. I told her that you come here at 7:45 or you are at the university. So like this. And if you sometimes if you want to leave early, then just put the time here. Otherwise, you need not to put. Okay. And each and every day you need to sign these things. And do not distract the class. If you have some kind of problem, you have uh, other things, you can go outside the class, but do not distract us and do not distract others. Okay. And you are elder people, so I need not to tell anything about that one. But sometimes one people last time, one student, actually he was listening some music in the class. So I just told him, just go outside and listen to music. You need not to sit here. Okay. If you miss 5 classes, then you will be failed. 5 unexcused classes. So better if you have some kind of things that you need to go for some doctor's appointment or some other things, just let me know why before. Okay. And I will suggest do not miss this class because one thing is related to another. If you miss too many classes, then you cannot get back to that course because particularly for the molecular biology, molecular genetics portion, if you don't know the replication, transcription, then you cannot go for the translation part. Okay, you need to know these things. So it is better not to be absent here. And quizzes or exams, if you miss one quiz or any exam, just let me know as early as possible. I will suggest that let me know before that if you have something because it is online sometimes. So if it is online, you will have 3 days time to finish that one. And within 3 days, you can finish it. But a lot of people are lazy. They don't want to do that. In such case, just let me know. Okay. And university attendance policy, we need to follow that one. So it is based on these attendance policy I need to submit to the university, and based on that one, your financial things will be determined. So be careful about that one, and always consult the canvas. This time we are starting the canvas because previous time we used the blackboard. This is the first time we are doing canvas, so we might have some kind of problem with the canvas, but just we need to go with the pace anyway. Because after June we cannot use the canvas, the blackboard anymore, and you cannot bring any person, any visitor into the class. Okay, and these are all policies. University policies are there in your syllabus. You can just go through that one. And when we will be doing the homework or any kind of project and other things, and this is very, very important right now, particularly for our part also. I am a researcher, I am a scientist, and we are doing the research and we need to present our data into scientific paper form. And this is very, very important right now. You cannot copy any other's work without giving him that thing like that. If you copy, we need to copy. We need to copy from book. We need to copy from others people's work. But we need to put the reference. But here, do not copy any other's project like that. Then you will get 0 points. Cell phones need to be turned off or in the vibrate mode. I will suggest put it in the vibrate mode because if you have some kind of emergency calls, then somebody can call you. But do not keep it open like sound. Do not do any texting or any other things here. If you want to do that one, you can go outside. You can go to do that. If you have any kind of emergency calls, just let me know and go outside and talk. Some kind of in case of emergencies. Okay, it can happen. It can happen to anybody. We are all human beings, so it can happen to anybody. So I'll suggest that one. I will suggest to bring your laptops. There is no problem. You can bring the laptops, tablets here, but use it for class purposes. If I see that one person is watching YouTube movies or something like that, you can go outside and watch that one. Okay? Because another person was doing the videos here in my class. So I'll not allow those things. You need to work in a group sometimes, particularly for the project. And there is a problem during this kind of group work is that suppose there are three people there working in a group, one person is totally absent from the universe. There is text, emails, phone calls. He or she is not receiving that one. Other two persons are working hard to finish the work. Okay. So in such case, what do you think? If you submit the project, I need to give the 3% the same marks. Okay. But in such case, is it good that one person, if he or she will not do any work, I will give the same percentage of marks to him or her? So in such case, I will suggest that make equal contribution in your project. Okay, each and every one. And based on your contribution, I will ask other members of your group that how much you contribute. So if your project will get 90 points, suppose like that, and if one person did the just 10% work on that one, he will get 10% of this 90 points. Okay. And when you are working in the group, particularly, it is also we are having this kind of problem that they are not submitting their data, or particularly for their data, other two peoples are waiting for a long time. So just think about these things because for you, don't delay other people's that they cannot do any work. And when you will contact me, just write this one in your subject line. So I can know that from where you are contacting me, from which class, okay? Because I have a couple of classes, so I cannot know that. So put the genetics, and we are meeting on Tuesday and Thursdays. So genetics, TR, and put your initial, just your initial. That's it. So we already have the canvas, and in canvas, I put only one module, and I'll keep just one module for the whole course. Okay. In that module, you will find all the files. Today, I just posted the chapter one slide also there. So all the slides, quizzes, and any other material, supplementary material, you will find in that module only one module. Okay. And this is your withdrawal rate that is from January 27 to April 3. And before January 27, there will be no record. And after January 27, between April 3, you will get a grade of W. After April 3, and if you do not come to the class, then you will get F. Okay. But I will suggest only two things. First thing is that consult your advisor for your financial matters if you want to withdraw. And second thing is that I will always suggest not to withdraw. When you are in the class, if you really don't have that much difficult problem here, then go through the course, finish the course as early as possible. It is not for me, it is for you. Because if you withdraw from class, I know a couple of people, they are withdrawing from the class and they are coming each and every time. After 2-3 semesters, they are doing the same course. That means you are wasting your time. Okay. If you have any kind of problem that you cannot understand these things, or you cannot go for the problems and other things, always ask. I am here to help you all the time. If you have any kind of problems, I'll help you. If couple of people are together having the same problem, because for genetics we'll do some kind of problems and logical things and other things. So If you have problem, just let me know by email that I cannot understand this part. If there are a lot of people, I will discuss in the class as a whole. If one or two people, I'll sit together and I'll do those things. Okay. And these are the university policies for the Title IX, which is for the sexual violence, dating or domestic violence. If you have any kind of these things, just let me know personally. Okay. Then I'll send it to you to Jennifer Red, and she will take care of the matter. We have the counseling services here. If you have any kind of problem, you can go there, contact them. We have Academic Success Center where you can get the free tutoring. And we have the library, already you know that. Other than textbooks, they are selling the laptops, iPads. Okay, so these are the official parts. And now our unofficial parts. It is our syllabus course. Okay, so already discussed about that one. So we'll discuss about the concept of genetics, molecular biology of the gene, and application of genetics, and we will have some kind of problems. Main idea is that to think logically and analytically. One thing is that for science, what I am getting all the problem all the times. Science is not literature. Okay, science is not literature. Science is conceptual. You need to develop your concept rather than just reading word by word. Reading word by word because your exam is not too tough. Probably I will go for the multiple choice questions and other things, and you can get it good marks there. So that is not the thing. But I want you to learn the subject. Okay. So develop your concept, and to develop the concepts, we'll go for some kind of problems because genetics. Suppose one person is colorblind and he married with one woman, normal woman, who is not colorblind. So how much you will expect that their children will be colorblind or not? This is the concept. You have to develop this concept. Okay. This is not that you can go through the books and you can find out some words and you can write that. You need to develop this concept. So all the times I will suggest just do some discussion in the class. If you, I know that all of you are here to learn this course. I do not expect that you know this course. Okay, theoretically this is true. You don't know this course, you are learning this course. That means you can tell the true questions, true answers, you can tell the wrong answers. Whether it is wrong or whether it is correct, that doesn't matter. Okay, just answer it. Sometimes a lot of things are coming out from the class discussion. I got very, very good results from that. We had a lot of good discussions here in the class. So we'll already talk about this one. And this is one thing that I would suggest just before coming to the class, go through the book already. So we have the idea, whatever it is. If you don't understand the things, that doesn't matter. Okay, just go through the book. Just or just one reading, it can send you the signal that I know this term. That is okay because genetics is full of lot of terms. Okay, when you have that idea, when you will discuss in the class, you will get the better concept for that one. Okay. Any question about that? Yes.
Can we record your lecture? Can we record the class, the lecture?
You can do that, but that will not really helpful because if it is visual, then it is okay because we need to do a lot of diagrams and other things here. So you can do that, but that will not really be helpful because a lot of people did that before and finally they gave up. Even in one class, I was there in one class and I was trying to record these things, but when I heard that one later, I thought no, because I need the visual things, what I am discussing in the board or writing in the board. But you can do that. That is no problem. You can do that. You can try it. Okay. One thing is that we have this schedule. This is a tentative schedule for us. Tentative. So sometimes We can have, generally, I am very much strict on the schedule, generally, but it depends on the class, that is environment. If sometimes I need to go for something a little bit more, I need to discuss a little bit more, it can overlap. Okay. So think like that. Sometimes I can finish a little bit earlier, particularly for the last couple of chapters, particularly this controller gene expression. Probably I'll not go too much details on that one because that is really difficult. So I might finish it a little bit early. This is tentative schedule. Sometimes that can be overlapped. So be aware of that thing. But generally, quiz, project, and exams will be on the same time. There is no problem for that one. And in the last class, we have the project presentation. Okay. So it is okay right now up to this. Any questions, any concerns about these official matters? Now can we go for the unofficial matter right now? Okay. So what is genetics? Again, so in this chapter, we will have the basic idea about genetics. What is the importance of genetics? Why do we need to know genetics? What do you think? If we don't know genetics, what will happen? Will there be any problem? Yeah, no problem. A lot of people don't know genetics. Yeah, that's it. Actually, the questions came fast that why the wolves look like their parents. That was the first question in the history of genetics. Why we look like our parents? That was the thing. Is there anything there? That was the first question. Okay. And that is the first thing what we did, based on what Mendel did that thing, that why the green pea plants are giving the green pea plants all the time. Why the yellows are giving the yellows all the time. Why? What was the problem there? That was the question first came. But later now we have a lot of things because we developed our science in very broad way, particularly for the diseases like cancer, AIDS, and those kind of things. That is very important for us to know the genetics part. Okay, because cancer is, I am a cancer biologist though, and that is the most deadliest disease in the world. And not only that, cancer is of different kinds. Previously, when I started working with cancer, I know that cancer means only one disease. Okay, during 90s, end of 90s, we know that each and every cancer is different. Each and every cancer, that means breast cancer is different than the prostate cancer is different than the liver cancer is different than the brain cancer and everything. Okay. During 2004, 2005, we knew that I was working with breast cancer and prostate cancer. We knew that the breast cancer means not only a single type of cancer, there are three different types of breast cancers, which are not equal at all. They have different kind of traits there. Right now, we know that there are six different types of breast cancers. Okay, and these are different. So that is the main part, that we need to know the genetics and molecular biology. Because what is the problem with the gene there? They are controlling that. You know, one news that very big news there. Angelina Jolie, she went from the double mastectomy. Okay, you know that news? Why? Don't you know that one? You need to know.
Why the double mastectomy? So that whatever the problem is can't jump from one breast to the other, so you just remove it.
Actually, the thing is that we have two breast cancer genes. One is called BRCA1, one is called BRCA2. This BRCA1 and BRCA2 mutation, if there is any mutation in this BRCA1 and BRCA2 gene, then there is a very good chance that person can have the breast cancer in the later. And Jolie's family, her mother probably had this BRCA1 mutation. That's why she was very, very susceptible for that one, that cancer. That's why she went for the double mastectomy to save her life. Okay. So these are the things we need to know about. That is the importance of genetics. We need to know all those things. We'll talk about a little bit of history of genetics. That is when, what was the concept, how the concept was developed, and main the fundamental terms and principles. We will learn a lot of terms. I will suggest you to write down all those things. Okay. Otherwise, after 4 or 5 classes, you'll be jumbled up. There are a lot of things. Gene influences our lives. How? I already told that. Gene influences our life. How? What do you think? Yeah, that is one thing. Another thing, in our regular life, I already told that.
Makes us who we are.
Yeah, that is that's okay. Then think about a very common thing. Not only for us, not a gene is not is responsible for only human. In each and every organism, they have genes, and we are working with those genes also. Bacteria, virus, they have genes. For AIDS, which is common mainly is human immunodeficiency virus or HIV. What is that one? They are incorporating their DNA into our DNA. So their DNA is attached with our DNA, and that is going to the T cells of our body. And we have different types of T cells. We cannot combat with the other immune things. That means we are losing our immunity. That is the main thing. So they have the genes also. And our agricultural products, we have a lot of things for our agricultural products. Like I told you, that is GMO, genetically modified organisms. So everywhere, everything is there in our life, all the things. Genes contribute personality, definitely. So because what personality you have, and that is from your father or mother, anyway. Generally, you can find a trend in the family of this personality. Personality is not only that one, also with some characteristics. Suppose bending finger, that is Mendelian inheritance, or hitchhiker's thumb, that is also Mendelian inheritance. You can have in the families. And it affects our susceptibility to many diseases. And this is one thing. This is a normal hand, and this is dystrophic dysplasia hand. What is happening here in chromosome 5, there is one gene here which is controlling for this disease. If there is a mutation for this gene, then the person can have this kind of arm. So it is like in cancer, there are a lot of genes which is in cancer, there are mainly two types of genes. One is called the tumor suppressor gene. Tumor suppressor genes means when it is active, it can suppress the formation of tumor. Okay, so that means if there is a mutation of the tumor suppressor gene, then the person can get cancer. Normally it is on, and if it is off, then person can get cancer. Another type of gene is called proto-oncogene. In anything related to onco means cancer. Proto-oncogene. It can transfer, if there is a mutation of this proto-oncogene, it can transfer to oncogene. And onco means cancer. When it is a oncogene, that means that person can get cancer. There are two things. One is controlling the formation of tumor. Another, when it is active, it can develop the cancer, develop the tumor. So, and not only that, some other things like that is chromosomal aberration, though, that is Down syndrome, Klinefelter syndrome, Cradichet syndrome. Those are also controlled by these genes. Okay. but the number of chromosomes, their structural differences, and other things. So it is related to many kind of disease and disorders. What is disease and what is disorder? Disorder means which cannot be modified anyway, which cannot be treated, which cannot be modified. That is called the disorder. That is Down syndrome. And those are called the syndromes mainly. And disease, sometimes you can treat that, but sometimes you cannot. Importance in agriculture. That is a high yield paddy. So we finally, this is the normal one when we used to get these paddies, this kind of paddies. But uh, what happened here, if they have high kind of irrigation, that is high water level, then these paddies could not survive because they have the low height. They couldn't survive in the high irrigation or high water level fields. So they modified the genes here and developed this kind of paddies which is long enough. So they can survive in that area. And we did it in various kinds of things. Particularly if you go to the, suppose, big bananas, big grapes, you can find, if you go to the grocery stores, you will find the GMO products. Okay, we develop those kind of things. We manipulated gene that they can produce this kind of things. This is for our benefits. Okay, and this is in the agriculture. We will discuss this one during biotechnology class. How we can modify those things. What is biotechnology? What is biotechnology? What do you think? Do you have any idea about
Bio
that?
was organic, right?
Yeah, bio is organic.
And then technology.
Technology is
Study of-
doing some kind of things with that one. Okay, okay, that's fine. But what is biotechnology? You have any idea about that?
Would it be like the CRISPR-9 or something?
CRISPR 9, that is okay. But what is that?
Some type of, I don't remember if it's a protein or not, but it's cancer.
Yeah, that is, yeah, related to cancer, that is okay. But suppose we are using insulin, the diabetic patients, they need to use the insulin all the time. Insulin is produced from the islets of Langerhans of our pancreas. Okay, so generally from our pancreas we can produce the insulin. How can we produce insulin in industries? It is producing inside our body. Either I need to take out the insulin by syringe, that how much is there, I need to take out the blood, I need to isolate the insulin from there, then I can give to another patient. Or we need to produce the insulin outside our body. How can we do that? Thing is that generally we have in genetics or in this whole course, we'll know about one term, that is one bacteria that is called E. coli, Escherichia coli. Okay, these bacteria, suppose this kind of structure, they have one outer gene. That DNA is present within the cytoplasm. Generally, DNA is within the nucleus of our body. But that DNA is inside the cytoplasm, outside the nucleus. Okay? This kind of DNA is called the plasmid. So what we can do that, we can isolate the human DNA and we can incorporate that human insulin DNA here in this plasmid, inside the bacteria. Okay? Now culture the bacteria, that modified bacteria. That modified bacteria you can culture within 1 hour, you can get 1 million of bacteria. Within 1 hour. Then they can develop into rapid scale. So from that culture media, you can find the insulin. This is called biotechnology. Cut the DNA from one organism, put it into the host, and do it, and do the culture there. This is called the DNA technology or biotechnology. Genes also develop in very important in development. This is actually Drosophila larva, the picture of Drosophila larva. And these are couple of genes which can develop different appendages. In this embryo, actually, In this embryo, there is no such things that from where they can develop their wings, from where they can develop their legs or other things. Okay, nothing is there. When we are also embryo, we don't have that kind of things. So the question is that we develop from a single cell, the zygote. That is a single cell. That single cell develops. So we have this cell. This cell develops like that. Now, which part will develop brain? Which part will develop kidney? Which part will develop lungs? Everything came from there. Simple thing. Which part will develop skin and which part will develop eye? They have the same number of chromosomes. They came from the same single cell. This is the thing. The certain genes are there which can determine that this cell can go for the development of eye. This cell can go for the development of brain. This cell can go for the development of lungs, such. Okay, we'll touch this part later a little bit. Couple of genes are there, and this is our model organism here. What is genome? You know this term also. You are very much familiar with this term, right? Genome. What is genome? What's sort of genetics? Genetics means a subject. Genetics is a subject. Genes means a part of DNA. So we have, how many chromosomes do we have? What is chromosome? First. Today we are doing some introductory work. What is chromosome? Just chromosome. What is chromosome? Where can we find the chromosomes? Nucleus of the cell. So, do we have only one chromosome or we have several chromosomes? Two pairs of chromosomes. Two pairs means how many chromosomes do we have? 22 pairs, 22, 26,
26.
23.
23.
22
Yeah,
46 total, 23.
26, 23,
23 pairs.
23 pairs.
There you go.
Okay, 23 pairs of chromosomes. And of these 23 pairs, 22 are called autosomes and one is called a sex chromosome, that is X or Y. Okay, why do we have pairs of chromosomes rather than a single chromosome? Paternal and maternal. Okay, so one came from father, one came from mother. That's why we have the similar chromosome in a pair. Do all the organisms have the same number of chromosomes?
No.
No. So in Drosophila, generally we find 4 pairs of chromosomes. Okay, but in human it is 46 chromosomes, or 23 pairs. And different other animals, even in some kind of fish, I found almost like 100 pairs of chromosomes. Okay, so those are these things. Now, do you think that each chromosome contain the same gene? So suppose we have 2 chromosomes here, one chromosome here and another chromosome here. Do they have the same gene? So each and every chromosome can have different types of genes, right? Does one chromosome contain a single gene or lot of genes? Lot. Okay. Now, just think about that. Can we cut the chromosome from the cell and take out that chromosome when we are doing the laboratory work, something like that? So we can take one cell. So can I? It is like a cut and copy in the paper. Just so you can cut the chromosome and take out that chromosome here. Do you have any idea about that? We can separate the chromosome. Can we? Hmm. Now, my question is that suppose I am taking one cell from here. I need to, can I isolate that chromosome by our laboratory techniques anyway? Just the concept. You need not to know that things, but I just wanted to know. Do you have this concept?
Yeah. Like cut. I saw with the stem of a bug, you can take like that eye chromosome and put it on like your leg.
We can take out the Y chromosome, that's fine. We can take out the I chromosomes, but my question is that we have 23 pairs of chromosomes. Suppose this is chromosome number 5. Can I take out the chromosome number 5 from a cell? If I need to do that, I need to do the cut and paste like that. So this is a set of chromosome. I need to cut the chromosome number 5, then I can isolate that. Can you do like that? We cannot do like that. Actually, we need to do some biochemical processes. We need to dissolve the chromosome materials there because chromosome is made up of DNA and histone protein. We need to dissolve the histone protein there. We can isolate the whole DNA. So from a cell, we can isolate the whole DNA from 23 pairs of chromosomes in a biochemical process. So we need to take the cells here, we need to add some kind of digestive enzymes there, then we need to centrifuge it. Finally, after putting the DNA enzyme and other things, we can find out a very white thing there at the bottom of the tube, and that is our whole DNA. DNA from 23 pairs of chromosomes. That means all the genes are there. This set of the whole gene is called genome because we cannot isolate chromosome, and that is called genome. Now clear the concept about genome, what is called genome? The whole set of genes from one individual is called the genome. That means for us, we have 46 chromosomes. All the genes of these 46 chromosomes in a total set is called the genome. Clear? So we have either RNA or DNA. What is RNA? What is DNA?
RNA is single-stranded, well, usually it has yours.
Okay, RNA is single-stranded, DNA is double-stranded. Another difference between RNA and DNA. RNA is very much volatile. It is producing for some times, then it is dissolved. You don't know that one, but I am giving you that information. But another major difference between DNA and RNA. Two major differences. Two. What is the full form of DNA? Full form of DNA. Full form of DNA. Deoxyribonucleic acid. What is the full form of RNA? Ribonucleic acid. So what is the difference between these two terms? And DNA has deoxyribose. So this is the difference. What is ribose? Ribose is not a protein. I think you have that idea that DNA is made up of three things. One is called the pentose sugar. Then it is attached, one part it is attached with the phosphate group, another part with the nucleotides. Okay, these nucleotides are adenine, guanine, cytosine, and thymine. 4 types of nucleotides we can find. And the main base, the strands, are formed with this sugar and phosphate group. This is like if it is a ladder-like structure, just like these things. This is made up of sugar and phosphate, and this is made up of nucleotides from both sides. Okay. This sugar, in case of DNA, is deoxyribose sugar. In case of RNA, it is ribose sugar. Okay. And another difference. This is one difference for the sugar.
RNA is uracil.
RNA has the uracil in place of thymine. Okay. So DNA has 4 nucleotides: adenine, guanine, thymine, cytosine. RNA has 4 nucleotides: adenine, guanine, cytosine, and uracil instead of thymine. This is the difference. And genome is copied during the process of replication. What is DNA replication? DNA replication means formation of another DNA from the parental DNA. Okay. So when the DNA is replicated, that means one chromosome is not replicated at all. All the whole set of chromosomes is replicated together. So that means the whole genome is replicated during this replication process. So what is evolution? Do you know the term speciation? And what is species? What are these three terms? Genus, species, and speciation. You have in your evolution class, if you had it before. What is species? There is a movie there, species, right? I didn't watch that movie, right? I didn't watch that. What is species? And what is genus? Did you do animal classification or plant classification before? Classification? Yeah? Then you can tell that. What is genus? What is species?
I think species is like species like how we're Homo sapiens, but like different. It's like
There is one term in whole classification system that is called binomial nomenclature. Binomial nomenclature. Nomenclature means naming. Binomial, bi means two. Nomial means names. We all have binomial nomenclature in our system, in our society. What is that? We have our name and we have a family name. Right? What is for the whole family, we have the same name, but we have different name for us. That is the binomial nomenclature. And they have the same thing. In each and every organism in this world have this binomial nomenclature system. They have one species name and one genus name. Homo sapiens. Homo is the genus. Sapiens is the species. Homo erectus. Same genus, Homo erectus. That is the primitive man. Homo erectus. Okay? So this is the binomial nomenclature system. Now the thing is that what is species? If you need to know the evolution, you need to know the term about species. What is the difference between species and species?
With species, they have similar genetic makeup.
Similar genetic makeup. Like, suppose we are human, Homo sapiens, and there is, suppose lion. Lion is Panthera leo, and tiger is Panthera tigris. They are from the same genus, Panthera, but Panthera leo and Panthera tigris. So, what is the difference between this leo and tigris?
Leo has the lion genes, and the tiger has the tiger
That means what?
genes.
That means what?
Um,
Be
like
very
they
specific.
came from a similar organism, I guess, evolved into two separate.
Okay, but what does it mean? Why did we give this name that they have the separate species? What is the difference between that? Separate species. Genus is a little bit higher. We need to go to the very basic level, that is the species. Species is a group of organisms which is reproductively isolated from other group of organisms. The only one term, reproductively isolated from other group of organisms. That is called the species. That means that if we mate them together, they cannot produce any offspring. Their genes are so much separated. Why? This is the first day. Today I am telling these things here. When we will talk about meiosis and mitosis, I will again ask this question and you will understand the difference between that one. Why it is possible that they cannot produce any offspring. Okay? So they are reproductively isolated. This is the most important term for the species. We came from apes. So ape was a different kind of species. And right now we are human, we are different kind of species. So we cannot produce any offspring through apes and human. There is no way. We are reproductively isolated, totally. And for this reproductive isolation, that is called the speciation, the formation of new species. So from one species, from apes, we are being isolated at different stage, different levels. That means we have some kind of mutation our gene first in the apes. So they are a little bit different from their primitive apes. Then that organism have more mutation. They have little bit different from their previous one. And after long generation, we became totally isolated. That is the mutation of our genes. There. This whole process is called evolution. So what is that? Certain change of genes. When there is a certain change of genes, but they are not really isolated, what is that called? And just give me one example based on our environment. There's certain change of gene, but still they are in the same species. They have some different characteristics. What is this called? In the broad way, when they are reproductively isolated, that is the formation of new species, then it is called evolution. Okay? But just certain change. What is the first thing? First thing is the mutation. And due to the mutation, there is some kind of certain change in their characters, behaviors, or some other things. What is that called? Right. Right.
I suppose it would be like Hulu and like 3 or 4 and then the next one.
Think about some kind of environmental factors. Think like the Tibetans. Okay, the Tibetans live in the 8,000 feet of height because that kind of altitude they can generate. There is a problem of respiratory problems. If we will go to that kind of altitude, we might have respiratory problems, but they don't have. They are the human. They are the human. Same thing. Adaptation. So that is called the adaptation. So first is the mutation of the gene. That mutation results in adaptation. If the adaptation survives for long generations, then there is a chance of speciation, and this whole process is called evolution. Clear? Very easy, right? Right? Okay. And for that reason, we can think that all organisms are thought to have evolved from a common ancestors around 4 billion years ago. We all came from one animal, only single cell animal. Do you know what is, who is our, that is humans, common ancestor? One human. We evolved from only a single female. All these populations, 7 billion populations in the world, we have only one common mother.
Lucy.
Lucy?
Lucy.
I don't know, but she has some kind of name. From Eastern Africa.
I think it was whoever was before Lucy.
Yeah, I think Lucy. The name. They give the name, but she is called the mitochondrial Eve. Mitochondrial Eve. And she is the common ancestor of all these 7 billion human population. Okay? DNA in the biosphere, we have tremendous amount of DNA and we are working on that. Okay, so we have the DNA in our nucleus. That's fine. Okay. Do we have any other kind of DNA in the organism other than nucleus? Nuclear DNA, other than nuclear DNA, do we have any kind of other DNA? What? Try to remember. You know everything, but just try to remember. Yeah.
Is it mitochondrial?
Mitochondrial DNA and And in bacteria, we have found one cytoplasmic DNA that is called a plasmid. In plant, we have another type of DNA. What is that? Rather than mitochondrial DNA. And I call the mitochondrial Eve. Why? Because mitochondrial DNA is transferred from mother to their daughter. It is maternal inheritance. So if you trace the mitochondrial DNA gene, then you can go to the ultimate mother. What is another one? Chloroplast DNA. Chloroplast DNA, mitochondrial DNA, plasmid, and nuclear DNA. Okay? Yeah, these kind of things. We already talked about this one. Transmission genetics means we'll talk about the Mendelian genetics and transmission, that is the inheritance pattern. Molecular genetics means that DNA, biotechnology, and translation, transcription, replication, all those things. Population genetics means evolution, adaptation, and all those kind of things. Now, one thing is that model genetic organisms. If I need to study the genetics, if I need to study the inheritance, do we study with human? What do you think? Why not?
Because it takes so long.
Takes so long time. So long means why? Why so long? What do we need to study? Try.
You want something more simple that takes a short amount of time to reproduce.
Time is what? Short amount of time is what?
Short amount.
I can give you short amount of time. That is not a problem. I can finish the class in a short amount of time. So,
But it reproduces.
short amount of generation. Okay. So within short period of time, we need to study a lot of generations. So that's why human is not a possible model organism for genetic studies. So we need to find out some kind of organisms we can produce rapidly and where we can find out a lot of generation in short period of time. Like pea plant. We can develop the plant in very easy way. And not only that, there are some other model organisms are here. These are what? Mainly short generation time, production of numerous progeny. We need a numerous progeny. But in case of human, if we study the human generation also in some way, but we can find out just 2 or 3 offsprings. But in case of pea plant, we will find the hundred of offsprings. In case of Drosophila, we can find the hundreds of offsprings. So we need to find out the lot of offsprings. Ability to carry out controlled genetic process. Controlled genetic process means we can control the process. In the pea plant, we can control the cross. That okay, I need to produce one kind of plant, just producing this yellow pea plant and green pea plant. We can control the cross. Ability to reared in the laboratory environment. We cannot rear human in the laboratory environment. But we need to rear those things in the laboratory environment because otherwise some other external factors can create the mutations in them. Then the genetic study is totally gone. Okay. The availability of numerous genetic variants. We need to find out such a species that where there are a lot of numeric, there is genetic variants of them. In case of Drosophila, we have white-eyed Drosophila, we have red-eyed Drosophila. They have different wing patterns, vestigial wing, normal wing, and a lot of things, gray color, body color, red body color, white body color. So I have different types of variants there. And accumulated body of knowledge about their genetic system. So where we can study the whole gene. And right now, this bacteria in E. coli. E. coli is a very, very common vector right now. This is called vector, in which plasmid you can transfer the other DNA, that is called the vector DNA. Okay? We know the whole genome sequence of this vector. So one is Drosophila melanogaster. That was the first model organism of genetics after pea plant. E. coli bacteria. And this is the most common right now in modern science. Model organism. And this C. elegans, we generally sometimes we are working with that one, but those two are main things. And this is another main model organism, not only for the genetic studies, but for different kind of things. Now the question is that if I need to study any kind of disease in humans, suppose I am working on cancer, suppose some people are working on diabetes, some people are working on AIDS. So we need to study in some kind of organism, right? We cannot induce cancer in human. We cannot induce AIDS in human to study that one, what is the going on there. We cannot do that. Ethically, it is not possible. Okay. So we need to study on some kind of model. Generally, we are taking this mus musculus, mouse model. Why are we taking mouse model? I have a lot of why questions, right? Why, what, when? That is the thing. Why do we need to take the mouse as a model for human studies?
Mouse can, isn't they keep reproducing, but it's
They can
closest
keep
to like,
reproducing, so why can't we take the
it's
cats
a
and all?
mammal. Or
It is
mammals.
mammal. That is the thing. We are mammals, mouse is mammal. And they have the shorter generation time, so we can study a lot of generations there. That is another way. They are very small size, so we can rear in our laboratory, so all the patterns can match there.
I know you like ingredient.
But the question is that I had this question in one saying. You work with mice, okay? So why do you say that this will work with humans? You work with mice. This, you work with mice gene, mice cancer. Why do you think that it will work on human?
It's,
I had to face this question. In the whole seminar, 100 or 200 people were there.
it's our chromosome, is our DNA kind of similar?
Kind of similar. 70% similar genetic structure we have between mice and human. That's why what we are thinking right now, what we are doing transgenic mice. We'll talk about that one later. Transgenic mice, that is the human gene we are transferring into the mice. Okay, I work with a couple of transgenic mice also. Transgenic mice, nude mice, we work with the nude mice means there is no immunity system in that mice. And one mouse is $600. One mouse, and you need to keep it in a very that is sterile environment. Otherwise you cannot study the AIDS because there is no immunity system. You need to develop the immunity there. So that's why we have the 70% similarity between mouse and human genetic structure. That's why mouse is a very good model for human studies. This is, we are not generally use the Saccharomyces cerevisiae, some kind of things. This is one, the zebrafish. Generally, we can study this is very useful for studying the change of skin color pattern of different human populations. Okay, we have very interesting story about the change of skin colors because we all evolved from one mitochondrial age. Then we went to different areas, different populations, different environments. Then for that one, we have two kind of things. One is called folate, one is called the vitamin D. These two are actually maintaining our incomes according to different environment. Then we have this adaptation, we had this, and that adaptation or evolution is going to generation after generation. Now tell me this one. What is the answer? One, two, three, four. Four things. A, B, C, D. A, B, C, D. I'll count the vote. How many of A? What do you think? A, B, C, or D? D.
D. I think
Okay. D.
D.
D. D. Anybody A? B? C? D is the correct answer. Okay. So other things is that we studied the genetics almost like 10,000, 12,000 years before, and we have lot of historical evidences about that one. But generally, it came from Aristotle, around 350 BC or something like that. So they have two different concepts. One is called the pangenesis concept, and another is called the germplasm theory. And we are in the modern world, we are following this germplasm theory. So what is that? In this pangenesis concept, during Aristotle or that time, they think that the genetic configuration comes from different parts of the body into that reproductive organ. Okay, they thought that this gene, they didn't have any kind of gene concept or anything. They thought that it is a fluid. All this fluid is coming or accumulated into the reproductive part of the body, and then it transferred to the sperm and ovum, and from there the other persons can get, or the offsprings can get, the parental characters. This is called the pangenesis theory. That is the liquid form of genetic material. They didn't know what is genetic material, what are those things. They thought this is a character, and the characters are there, every part of the body is accumulating there, and it is transferring there. That is making a package, and it is transferring. Okay. Right now we know from the Mendel, actually Mendel didn't know the term gene. After death of Mendel, almost 10 or 20 years after the death of Mendel, the term gene actually coined. He turned the things as factor. So there are some factors, but that is a solid fact. It is not the liquid. That is a solid factor. It can go or it cannot go. Not only that, that factor can be either dominant or recessive. So some person can have, so according to pangenesis theory, if there is a red flower or there is a white flower, according to this one, they can be mixed together, they can form the pink flower. Okay. But in case of Mendel's theory, that is called the germplasm theory, that when there is a yellow pea plant and then a green pea plant, or one is a tall pea plant and short plant, we can found the tall plants in the first generation. There is no short plant here. Where is that mixed? No, they have not mixed yet. Because in the second generation, we'll find the short plants. So they are different kind of solid things. They can be isolated in this way. And from that concept, we got the dominant and recessive things. So this is the accepted theory right now. Okay. Just need to know about this one. There are a lot of things to remember, but just think about these things. The one is what is called the pangenesis theory and what is the germplasm. Because there are a lot of things to know, and when we'll go to the 5-6 chapters later, then it will be nothing. Okay. And these are the actual timeline for the genetic studies. First, the father of genetics, Gregor Mendel, who coined the things of principles of heredity, how the factors are transmitting from one generation to another generation. That is called the heredity or inheritance. Okay. Second things, Sladen and Swan, they proposed the cell theory. During Mendel, he didn't know about the cell also. So they were the first person who proposed the cell theory, that we are made up of cells. Each and every organism are made up of cells. Fleming first coined the term chromosome. They found that in the cell, there is something which is called chromosome. Chromosome means color. So when they stained the cell, they got a thread-like structure which is colored blue. That's why they called the term chromosome. But they didn't even know that where the Mendel's factor is. Is it within the chromosome? It is within the cell. They didn't know that. During that time, it is around 1900 to 1904, something like that. And Mendel is 1870 or something. Okay. Then Darwin proposed the theory that is called the evolution. What is called evolution? Okay. Weismann proposed the germplasm theory that no, it is not pangenesis, it is not mixed. Mendel didn't know that, but he proposed the term, but he actually coined the term. It is called germplasm theory. It is coming through the gametes. Certain propose the genes are located on chromosome. This is the first time they have found out where are the genes, where are the Mendel factors. They found out, okay, the Mendel factors are located on chromosome, not within the cell, not within any other areas. That is on the chromosome. Even that time, DNA was not discovered. DNA was discovered around 1960s. And this is the story of 1904, 1910, something like that. This is all about that, and this is modern genetics. This is the DNA sequence. What we are doing that, we need to take out the DNA, we need to put different nucleotides there with the fluorescent tags. Then you need to run on the machine, and that machine is giving up these graphs. Based on this graph, the high peak, this is blue, this is C. This one is high peak, is A, that is, it is adenine. But we need to read this graph, then we can find the sequence of the gene or whole DNA. Okay? So this is all about our introduction of genetics. I think you have a very good concept of that. Do you have or not? So from the next day, we'll go through the principles of that is the inheritance, principles of inheritance or hereditary factors, and I'll give you some kind of problems there. We'll work in the class about the problems. Without problems, you cannot understand genetics. And the very, very basic thing is that do not treat this subject as a literature. Treat this subject as conceptual. Okay? Read the diagrams rather than reading the language. Read the diagrams. A diagram is saying a lot more things than you're reading. Read the diagrams, where the arrows are going, what are the things there. Always read the diagram. Okay? In each and every biology class, I am telling this. You need not to read the whole things there. Just read the diagram, try to memorize that thing, you will have the clear concept of that. In your whole life, you will not even forget that. Okay? So this is all for today. We'll meet next day at the same time.